The next session, the environment. The speaker is Professor Krishna Reddy from Chicago, and your turn will be, you have 45 minutes and your turn is this lecture.
Yeah, good morning, everyone from Chicago. So I know it's afternoon for you. I'm very glad to participate in your symposium. I was in Budapest just last week. I really enjoyed Budapest. Unfortunately, I didn't have much time because I was with my family. So I look forward to coming to Budapest again, and it's a very nice place. I really enjoyed quite a bit. Um, yeah, my presentation topic is on landfills, innovative biochar-slag-soil cover system for zero emissions at landfills. I know I saw the program. You have fourth part of this symposium talks about biochar. So hopefully those who are working on biochar may find this presentation useful. Before I get started, I want to thank Dr. Antal and Dr. Emery for inviting me to participate in this symposium. Unfortunately, I could not be there in person, but I hope my online presentation can connect us and then we can meet in the future in person. I want to thank my graduate students who really did the research that I'm presenting, and also funding for this research is provided by National Science Foundation and industry partners in the US. So Chicago, if you know US, this shows where Chicago is. I really want to invite all of you to visit Chicago whenever it's possible. Chicago is a beautiful city. Of course, it's relatively different than Budapest. So I want you to invite you to come to Chicago. And the last two pictures you see here, they are my university campus. So it's right next to downtown. So you can walk around the downtown and visit my university as well. So at this university, I am in charge of these two labs, Sustainable Engineering Research Lab and the Geotechnical and Geoenvironmental Engineering Lab. So we do a lot of research on environmental remediation of soils, sediments, groundwater, and stormwater. And we do research on waste management and landfill engineering. And we work on life cycle assessment and sustainable and resilient engineering. And we also work on traditional geotechnical engineering topics. So you can find more information about our projects on these websites. It's always challenging to pick a topic to present because I like working on all these topics. So I chose one on biocovers. I know your group works extensively on closed landfills, and perhaps this topic would really be of interest to you with your ongoing research. So over the last 25 years, I have pleasure of working with researchers from all over the world. I hope to see some Hungarian researchers in my group as students or visiting scholars. I also work, with a lot of international collaborators. Hopefully, we can also work together on some of the mutually interesting projects. Whatever research that we have done in the last 25 years has been published in these four major books and a lot of journal papers and conference papers. So you can go to this website and get all that information. So in my presentation outline, I just want to talk about landfill gas emissions problem. And then how can we solve this problem using a new cover system? We call this one as a biogeochemical cover system. Basically, it uses these three materials, biochar, slag, and soil, and I'll show you how these materials can be used to basically eliminate the landfill gas emissions. So our research methodology and results, and then end with ongoing and future research and few closing comments. I hope you can hear me well and you can see my slides well. So unless I hear any interruption, I will assume that everything is going fine. So global solid waste generation, this is a huge problem. No matter which part of the world you are looking at, we are generating a lot of solid waste. You see here, uh, in North America, where I am in, you can see we generate about 289 million tons of waste per year. This is per year. And, even per year amounts are increasing. So you can see Europe and Central Asia, 392,000 tons per year. So you can see large amounts of waste are being generated, and these are huge quantities, right? Millions of tons per year. And you don't know what to do with the waste that you generate this year, and you keep accumulating more waste every year. So it's a huge problem that we are facing. And with the population increase and with our lifestyle, this waste, unfortunately, keeps increasing. And the projections show that we could have globally, by 2050, 3.4 billion tons of year. We're talking about only the solid waste. There are many different types of waste, but municipal solid waste is the one that we generate in the houses, in community entities, restaurants, institutions, and so forth. So this is a lot of waste. And so what are we doing now with the waste? Basically, you have this open dumping or landfilling, composting, recycling, and incineration. So if you look at what is going on, for example, in North America, in the US, we have mostly landfills, engineered landfills, and then we have some recycling and then some incineration. So you don't see too much of composting, no open dumping at all. South Asia, I'm originally from India, so you can see most of the waste is dumped illegally, open dumping, which is really bad. You have only 4% engineered landfills, 16% composting, only 5% recycling, which is really, really bad. In Europe and Central Asia, you see the balanced activities, but it seems like, in some countries, you still have open dumping, you have engineered landfills, in composting, recycling, and incineration. So you can see, what is going on. So it's sad to see open dumping still taking place in some countries. The best way is to at least to use engineered landfills so that environment is protected. But you need to think about waste management from integrated way. Of course, you want to collect and sort, and you recycle the materials as much as you can. And then you do the biological treatment, then thermal treatment, and the last option would be the landfill. So depending upon the geographic location, depending upon the country, depending upon the situation locally, you can adopt a combination of these things. As the geotechnical and environmental engineers, we deal extensively with landfills. So this is the topic of my presentation. So landfills are engineered structures. You have a bottom liner and you have a side liner. You have a leachate collection and removal. You have leachate collection and removal system, and then you pile up the waste and you put the final cover. And the design of these landfill components is very well established. And in the US, we have been having these engineered landfills for decades, and there is a lot of monitoring data. It shows that the landfills as designed and constructed, operated, are protective of the subsurface environment. So everything is good. but we cared about leachate, and then we have leachate this landfill this bottom liner and side liner and leachate collection and removal system so that we don't have accumulation of leachate at the bottom. So we have taken care of the leachate. But in terms of the, when your waste degrades, right? Because it has organics, it degrades and it generates gases. Basically, you have methane, CO2, H2S, and non-methane organic compounds. And a lot of times we have these gas extraction wells put in. Basically, you apply the vacuum, you suck it up, and then, a lot of times we use the landfill gas for energy generation purposes, which is really, really good. But the problem is, these gas extraction wells are not spaced closely. They're spaced far away, and the areas that are not within the influence zone, they come up to the cover, and from the cover they diffuse and into the atmosphere. So this is a huge problem in the U.S. Even though we have a final cover system, gas collection systems, uh, significant amount of gases are emitted into the atmosphere. We call this one as fugitive emissions, right? So we have methane is the third largest source of methane in the U.S. So if you are thinking about methane is the greenhouse gas, and like if you are thinking about controlling the greenhouse gases to address the climate change issue, you could do something about these emissions in the landfills that can make a lot of difference. So you have a CO2 emissions, and the problematic gas is the H2S, because the neighbors will complain so much because it smells. Methane and CO2 do not smell, so neighbors, don't know about those emissions. But H2S, even small amounts, it smells so bad. There are a lot of protests by the neighbors. And these non-methane organic compounds could be all kinds of air contaminants, air pollution issues, and that could harm the health of the people exposed. And we're talking about this problem with engineered landfills. Think about problems associated with the dump sites. As I mentioned, they are existing in many countries, and you are directly emitting all these emissions into the atmosphere, which is a huge problem. So what we came up with is this biogeochemical cover system concept. So we have an engineered landfill. We have a bottom liner, side liner, leachate collection and removal system. You have final cover. You have gas extraction systems. But as I mentioned, these gas extraction systems are not taking out all the gases that are generated, and they are being transported to the cover system. So we have to do something to the cover system so that we can control the fugitive emissions. So what we said is, instead of using the standard cover system, we want to use this new cover system. So we call this one biogeochemical cover system. So let me explain this one. So you have a landfill at the waste at the bottom. It is degrading. You are generating the methane, CO2, and H2S. And then we are suggesting to have a biochar amended soil cover, biochar amended soil layer. And what happens is, in this biochar amended soil layer, you have a methanotrophs. Basically, these are the microbes. They can convert methane into CO2. And then on the top of this biochar amended soil, we're suggesting a slag layer, basically steel slag layer. And then that can convert all the methane that is converted into CO2. That CO2 can be sequestered. And the CO2 that is nothing is happening in the biochar amended soil, that could come into this layer, and that could be sequestered. And H2S also could be sequestered using these reactions that I showed here. So if methane is converted into CO2 and all the CO2 H2S is sequestered here, you practically don't have any emissions. So that's why I call it as zero emissions. You can see the mechanisms of what is happening. So you have a biodegradation, anaerobic biodegradation that is generating methane, CO2, and then in the biochar amended soil, you are getting this methanotropic bacteria or microbes that are oxidizing the methane. And here you have the sequestration, carbonation reactions that's happening. So let me explain these two layers, biochar amended soil layer. What is biochar? As I mentioned, your program, third part of today's program is about biochar. I didn't know about it, but it's really, good topic to address. So biochar is basically you take any organic mass and then put it in biopyrolysis or gasification unit. So basically, you're heating up that biomass at high temperatures with no oxygen present. So you get these bio-oils, gaseous fuels. So basically, you're creating the biofuel from the biomass, which is really, really nice. And a lot of chemical engineers, mechanical engineers are trying to convert the waste into biofuels. In this process, they consider this solid waste that is generated, they consider it as a waste, and we call it biochar. And it comes in different forms, physical forms, depending upon where we call this one feedstock, and then depending upon the pyrolysis process, you can get this biochar. Basically, it's like dark gray or black looking, and the physical form could be pellets or like a little bit granular. You can see that. And it has a very nice properties. It has a high surface area and internal porosity, high water and nutrient retention, higher resistance to biotic degradation, ability to absorb gases, and it can enhance the microbial colonization, right? So it's an inert material, but it has a lot of micro porosity, and microbes like to reside in those spaces and have higher activity. So this biochar, extensively used in agriculture. Basically, they go and spread it on the land, and then they till it, and it is shown to increase the agricultural yield. And it started to be used in environmental remediation of soils and groundwater. And then, we were the first one to think about why can't we use this one in the final cover of the landfills to sequester the gases, right? So one thing I want to mention right up front, people use the term biochar in general, but all biochars are not the same. So I have a paper and a presentation that I talk about. Depending upon the combinations of your feedstock and the pyrolysis processes, you can get different types of end product. Biochar could have different properties. So I'm showing you, the I generally don't prepare biochar in my lab, but I go to commercial vendors. There are many commercial vendors in the U.S. So I got the samples. So these are the different types of biochars I got, and then we did the characterization in the lab. Okay. And one thing you have to really worry about using biochars is about the contamination that they could have. These polycyclic aromatic hydrocarbons, these are toxic organic compounds. And then the metals, there could be toxic metals also in there. So you have to really look at it, okay, how much they have it. So if you have a lot of contamination, in biochar, biochar is basically, from waste derived. Any waste derived product or waste, you need to really worry about this chemical contamination issue. So the point I'm saying is that you need to use the biochar that has low contamination or no contamination, or at least the contamination should be less than acceptable values. Now the question is, if we add biochar to the soil, how could it help, right? How could it help? So to investigate that one, you can do some batch experiments. Batch tests are very simple experiments, wherein you take some of these small glass cubes, and then you put the material, and then seal it and put the gas in there and monitor it, what is happening to the gas with the reaction with your material. But these don't represent real world landfill cover system. So we create these columns, and then we can inject the gas from the bottom. These are like a simulated cover systems, and we can put the gas from the bottom. As it transports to that, we can measure the concentrations, and we can measure the gas coming out, and then we can analyze the results. So we call it batch tests you do first, and then column tests, because the column tests involve a lot of work, and then there are long-term tests. But batch tests are very quick to run and see, if you have any benefits of using the biochar. So I'm showing you some typical results. For example, if I have a soil itself can have, methanotropes. It's not, every soil, if you characterize it, they will have some methanotropes. So this is the dark solid symbol. You can see here when you do the batch test, it's showing how much of methane is uptaken. Basically you are oxidizing with the methanotropes in there. So you can see a lot of things are happening with time. You can see a lot of methane is being converted into CO2. But if I add 2% biochar to the soil, you can see there is not much difference initially. The later on, once the microbes acclimate to the biochar, you can see the rate of removal increases. And if I add more biochar, 10% biochar, you can see again in the beginning, this acclimation stage, and after that, you can see it will take off. The initial acclimation period is important. If you don't want to have that initial acclimation period, you can sometimes, activate the biochar with the methanotropes right from the beginning and put it in the soil. And then you can see there is no lag time, and you can see this is the 2% methanotrope activated biochar in the soil. You can see there's no lag time, it keeps on going. And if I have a 10% methanotropic activated biochar, so you can see it goes up. So with these simple experiments, you can see, it takes time, right? This is a biological process, it's not a chemical process. It doesn't happen very quickly. So you really have to have patience to monitor for a long period of time. And this can go on perpetual because there's not like a limitation on the capacity. It's just a biological process that keeps happening. So you can clearly see the benefit of, if you have a soil, you can get the methane oxidation conversion into CO2, but it's very slow. But if I add biochar, we can really increase that to higher capacity and higher rate. So that can be found from batch experiments. But you need to do column experiments. As I said, column experiments are the ones that simulate the landfill cover and the gas transport through it. So we did a lot of tests. So with the soil control, you do with the soil alone. And then we did with 2% biochar layer, 10% biochar layer, or 10% biochar layer to the entire column. So if you do the soil versus, let's say, 10% biochar, if you look at it, see the concentration profiles of methane and CO2, they are not much change. But here, when you add 10% biochar to the soil, you can see significant reduction in the concentrations of this methane and CO2. And also, the methanotropes are, need oxygen. So we also monitor oxygen. So you should not have any, limitation on oxygen, because oxygen can enter from the top into the soil. So you're not really providing that oxygen, but that's why even the cover systems are near the surface. So there's always ingression, and this can happen. So if you are interested in looking at the methanotropes, how the quantity and the types of methanotropes that you have, you can do the detailed microbial characterization. So, in good old days, it was really hard to do these things, but these days, it's very inexpensive to do these tests. So you can take out the soil that is exposed to methane and do this characterization. You can find out exactly, how much amount of methanotropes that you have and what types of methanotropes you have. So the general result of, reduction in methane. But okay, we are saying that it is due to, microbial oxidation. But then, what microbes are really doing the job. If you want to learn the biology, then you can do this microbial community assessment and understand which specific, methanotrope. There are many types of methanotrope microbes, hundreds of them. So hundreds or even thousands of them. But so which one is at play, you can look at it. So doing the batch test and column test in the lab is not enough. You need to go to the field and test it if it works in the field or not, because you cannot really simulate the natural environment in the lab, no matter what, right? Weather, rainfall, temperature changes, and other things. So we went to a landfill and then we created these 4 different profiles. The first one is just the soil, is basically in the top. And here, this is the soil profile, a soil liner with a 2% biochar amended cover here. Instead of whole layer, we put only like a small layer. And in this one, you can see, we try to put like, okay, can you put like a small layer of biochar, not amended? And then here is the situation where we had 10% of the soil. So basically, we are constructing these covers in the landfill. So we went to a landfill close to Chicago. The waste was filled. They put a temporary cover on it. So we are taking out that temporary cover, and we are making a trench. And then we are backfilling with some gravel material. And then we put the soil, and then we put a layer of biochar, or we will amend the soil with the biochar and put that layer to simulate the profiles that I mentioned. And then once you at the ground surface, we also place these gas collection tubes at different depths so that we can collect the samples, as you see here. So we can, actually use the syringe and collect the gases. You know what is happening at different depths. And then, of course, you can also do this chamber methods to measure the gas emissions too. So these are the completed things, and then we go once in a while and then collect the gas samples from these pores, and also we use the static chamber method to look at the emissions. And then you can look at the results. You can look at for the soil alone, you can see the concentrations don't change, but once you have a biochar, you can see the concentrations reduce significantly. So again, in the field also, it is demonstrated that the biochar presence can really, eliminate the gases. So we looked at the biochar amended soil. And then let's move on to this, the top layer, slag layer. The slag is is a waste product that comes from the steel industry. Near Chicago, we have a lot of steel plants, and they create different types of slags. And for coarser slag, there are many markets. They actually sell it. But there is a finer steel slag that nobody wants to use it. You cannot have a good geotechnical application. So the piles of these materials are there at these plants. And only way to get rid of these is to send them to a landfill. So our idea was, okay, this is the waste material, and this industry is ready to give for free. Can we use this material as a landfill cover material? So we got this steel slag from 3 different plants. Basically, this is one plant. We got it 2 times. And then there's a Riverdale, there's another steel plant. So we wanted to see, and then we characterized this steel slag. The specific gravity is, of course, very high. And then hydraulic conductivity is close to 10 to the power of minus 3 to 10 to the power of minus 4. And then one important thing that you have to keep in mind is the pH. pH is very alkaline. And another thing important to look at is in the reactions, as you saw, you need a calcium. So you need to look at what are the mineral sources that could provide calcium for carbonation reactions. And if you have H2S, you need to have an iron source. You need to look at what kind of minerals that you have. So you really need to do this analysis before recommending any slag for these kind of applications, because you need to have enough calcium and enough iron so that they can react with CO2 and the H2S. So then that's what. So again, To check whether this thing happens or not, you can do simple batch experiments, or you can do the column experiments, right? Small column experiments. You don't have to have a large column. You can do the small column for this material testing. So if you do the batch test, basically we are taking the, in the batch test, basically we're taking the slag and we're injecting the CO2 and measuring the concentrations with time. So if it is sequestering, CO2 concentrations will reduce, right? And then we can calculate with time how much is CO2 is removed. So this shows the CO2 removal for the 3 slags that we looked at. So you can see it removes quite a bit, right? And we injected CO2 and methane mix, but nothing happens to the methane. Obviously, in the cover system, the biochar amended soil would have taken care of all the methane, and then only CO2 would be coming into this layer. One important thing about the efficacy of this material is you need to have some moisture. If you don't have any moisture, for carbonation reactions, they happen in the solution form. So we looked at 10%, 15%, 20%. So at least 10% is good enough for the carbonation to take place. So again, you need to look at it, since these cover systems are on the top, when it rains, the water gets into it and the moisture is retained. So you have to check if that moisture retained is enough for these carbonation reactions or not. So the point is, you cannot expect a dry material and have these reactions take place. So and you can see the column experiments. You won't see anything coming out for many pore volumes. You need 100 pore volumes to see any kind of CO2 coming out. So the material will exhaust, not like a biochar amended soil where biological reaction can keep on going. But in this one, the capacity is limited by how much carbonation that the material can take. So this slag will have a finite life in terms of its reactivity. And you can see how much is the cumulative CO2 is removed. And you can look at these profiles and you can understand the mechanisms of what's happening at different stages. You have these different minerals providing the calcium for the reactions, right? So when one easily available calcium gets exhausted, then the calcium has to come from the other mineral. It may not come as quickly as the first one. You can do interesting understanding of the mechanisms, explore the mechanisms that are happening. And then H2S, simultaneously, we had injected CO2 and H2S. You can look at it. CO2 removal, also H2S removal can also happen, which is great thing because, you can avoid the odor problems at this landfill. So substantial amount can be taken out. And the other thing that I'm showing here is the gradation. The CO2 as is the one that we got from the steel plant as it is, but you can separate them into fine fraction and coarse fraction, and you can see the finer the fraction, finer the slag, better it is, which is good because coarse slag can be used for road applications as aggregate and other things. Fine fraction is the one that does not have any uses right now. It's considered as a waste material. So, saying that finer slag is good for this application is like, very good news for this, the producers of the slag. And then we tested, biochar amended soil and slag separately. Now, in the biogeochemical cover system that we are proposing, we want to have both of them, right? So we try to test it. So basically, this is the soil-only layer. This has the 10% biochar at the bottom, and then you have a slag on the top, right? So this sand layer, small sand layer, we put in because we can measure the concentration that is coming into this. So we can see how much is coming from the biochar layer, amended biochar layer, biochar amended soil layer, and then how much is feeding into the slag layer, and we can measure what is coming out. So the sand layer is not necessary, but we just did that one for measuring the concentrations. But the whole idea is this is only soil. This one is 10% soil layer at the bottom, 10% biochar soil layer at the bottom, and then you have slag on the top. And we looked at here 5% activated biochar soil, and then slag as is. And then we looked at one test with 10% activated biochar and slag as is. And then we tried to mix slag with sand. So this is a different rationale for selecting all these things, but you can look at it. This is the soil only. This is the soil, biochar amended soil, and the slag. So this is, would be my biogeochemical cover system. This is kind of a conventional soil cover system. And you can look at the results. Basically, we monitor the concentrations over a period of time. You can look at it. So the first one is the soil control layer. You can see the concentrations of methane is the red, and then blue is the CO2. They're decreasing, but at the top you have some emissions. But if I have a soil with 10% biochar and the slag on the top, you can see the concentrations decrease, and the CO2 concentrations, you can see this, the blue is CO2, nothing comes out. And this one, there is small amounts of methane emissions. But if I use these activated systems, you can see the concentrations of methane decreasing and CO2 increasing in the biochar amended soil layer. That's what we wanted to see. And once that CO2 comes into the slag layer, you can see, it reduces. So in this profile, you can see nothing is coming out. Concentrations at the exit point are 0. So that would be like a best scenario you can think about it. I'm not presenting it here, but for, we presented in our papers, it's a good idea to look at which methanotropes are doing the job, right? So we know the results, but if you really want to understand the microbes, you can do this DNA and then look at, so we looked at this one, soil control, 10% biochar, those 4 columns. And we took out the samples, we extracted the DNA, and we did the PCR analysis. And you can see these types of methanotropes. You see that the one which in bold, those methanotropes, those particular methanotrope microbes are really, consuming the methane, oxide in the methane. So then, okay, that's good, very positive news. So then you need to do out in the field, like what we did for biochar amended soil. We wanted to do the same thing for this, biogeochemical cover system, but we just got COVID during that time, and we couldn't go out to the field. So we created a large scale tank in the lab, and we simulated this one. So you have a biochar amended soil at the bottom, and then slag on the top, and top soil layer is basically some vegetative layer. And then we were injecting the gas from the bottom, and the gas goes to the biochar amended soil, and then goes to the slag layer, and it comes out. And the top, we can seal it off and collect the gases, or we vent it into the fume hood. So this is how those 2 tanks looks like. This is just a soil cover tank, and this is a biogeochemical cover system. And then the cover is simulated in this one. We are injecting the gas from the bottom, and then we are venting it into the hood. Or when we want to measure the flux rate, we can just take out these covers and then put the other cover and then measure the gas emissions. So we did these tests on the different phases. We changed the inlet concentration, or we changed the methane flux, because it's not easy to do multiple tests, right? Same setup. So we can vary the flux rates and then duration and look at what happens in the system. So good thing is, we had that transient data, but also at the end of the testing, we were able to exhume the samples and then collect the samples and do the testing, to look at remaining oxidation capacity and carbonation potential. So because the tank is really large, and you can collect the samples, and you can get the spatial variation also in this one compared to simple cylindrical setup. You assume this is a 1D and uniform conditions, but here the gas flow could be changing spatially, so you can get that interesting results. So to make simple very easy, we whatever happens, whatever happened, whatever we found in the batch and column experiments, were confirmed in this large scale experiment. So different phases, we could see the methane removal and carbon dioxide removal and so forth. And also we did the microbial characterization, and we found out that type 2 methane is the one that was dominant in many of these column experiments. Then we wanted to answer this question: is this cover system sustainable, right? So we did the life cycle assessment for typical soil cover versus the biogeochemical cover. So we looked at the materials, where the materials are going to be coming from, and both materials, for biochar, it's a waste material, so no emissions involved in making up it. And then the slag, the slag is also like a waste material because it's coming out of the steel manufacturing. So you are utilizing the waste material, so there is no environmental footprint of making them. And the only thing you have to worry about is transport of this material. So you are applying at one location, but how closely these materials are available. If you have to transport these materials from long distances, then emissions associated with that, makes these systems unsustainable. So just the typical results, I'm talking about soil cover versus BGCC. You can see a lot of carbon sequestration in terms of the global warming. So both covers are going to benefit, but the BGCC will have a huge carbon sequestration potential, and you can also see a lot of benefits of BGCC in other environmental impacts. So the question is, we tested the biochar. We tested one particular type of biochar, basically waste pine wood based biochar, and then that may not be available, right? So biochar can be made from different types of feedstock. So we looked at biochars from different types of feedstock and tested it. And the other question is, steel slag may not be available, at the landfill that you are working with, or not available even close by. So can we use other types of materials instead of slag? So we did testing with all these different materials and alternative biochars. As I told you, depending upon the walnut shell biochar is not as good, but you see rice husk. Rice husk is easily available everywhere. If you use the rice husk, that will give you biochar excellent, qualities to sequester. To oxidize the methane. So carbon stock. So, don't take it for granted. Just because you have one biochar, you want to use it, it may not work. But you need to check which type of biochar is good, and if you have that feedstock close by. And one advantage of biochar is you can scale the manufacturing. You can scale it to small level. You can manufacture at the landfill site. Which is kind of very nice. And then we did the Biochar-Slag, but fly ash is available a lot of places. Cement kiln dust could be available. Concrete waste fines could be available. So we tested these things, and they have different degrees of carbonation potential. So you need to really check it. So we found out cement kiln dust has a really, really good capacity to carbonation. So we did the testing with these things, just like how we did with the slag. So the points to take away is, we are calling this as a new biogeochemical cover system to remove those methane, CO2, and H2S. So practically, we are going to have a zero emissions. And it contributes to decarbonization and climate mitigation. And it's practical. We're not looking at a very fancy materials, fancy construction. These materials are easily available and we can use it. And I didn't show you, but we did the testing under varying moisture contents, varying temperatures, from low temperatures to high temperatures, and we found out that the performance, of course, changes depending upon the environmental conditions, but there is always positive results. So it's resilient to climate conditions. And then, biochar is from waste material, Biochar-Slag is from waste material, CKD is also waste material. So we are using the waste materials for beneficial purposes. We are avoiding the disposal in landfills and also avoiding the stockpiles at the places where these are generated. And, in the context of sustainability, we are resource conservation, right? We are eliminating the waste, and we are avoiding using sand or clay or, natural materials. And it's economical. As I told you, biochar is easy to produce on site with waste biomass. And Biochar-Slag, the steel plants are desperate to get rid of them, and they are providing for free. Only thing is, transport costs. And, I showed you some life cycle assessment results. It's sustainable. So we want to test this one in the field. And, you guys work on landfills. If you want to test some kind of systems like this one, because I know you guys work on closed landfills for energy purposes, solar and other things, and at those closed landfills, if you have a problem of gas emissions, so you could recommend some of this kind of a passive system, and gases are fugitive gases are. Of course, I'm always for collecting the gases and using it for energy recovery. Don't take me wrong in that sense. But any fugitive gases that you cannot capture, so this is like a nice sustainable passive system to take those out. And we did this integrated resiliency sustainability assessment, and we need to develop some practical design guidelines so that industry can use it. So I went through very quickly, but we have published a lot of papers on this research project. So you can find all those on my website. These are my websites, and then this is my email address. You can connect me on Twitter, Facebook, and LinkedIn. But I will be very happy to answer any questions, anytime in the future if you want to undertake any of these kind of projects. So that's my presentation. Thanks everyone for listening to me, and I can take on any questions that you have, or feel free to email me your questions. I can answer later on.
Thank you for your very interesting lecture. And I'd like to ask the audience online and in the room, do you have any questions? Somebody else? I cannot see it. Then please go to the microphone and.
Okay. Just thank you very much for your nice presentation. I was just wondering if you could comment on the sort of design line of a typical 300 milliliter stamp layer. Certainly, it will have a limited capacity to sequestrate CO2.
Yeah, so that's a good question, and that's the question we always are asked when we talk about it. So as I mentioned, that biochar amended soil has no limitation on the capacity, right? So it's a biological process.
Indo-European, mert tényleg jó, nyilván. Igen.
All right. So the biochar amended soil, in terms of its oxidation potential, that can go on perpetual. But the limitation is on the slag. Slag has a limited capacity. So we did calculation of the lifetime of it. So it depends upon how much is the CO2 emissions that you're getting from the landfill. So as I told you, we are basically addressing the fugitive gases, not like all the CO2. And for a typical landfill, we found out that it could last for 10 years or so. Again, it depends upon the emissions, right? And also think about this one. We don't have these emissions perpetually because there's a limited amount of organic matter. Once it degrades, you don't have any more gases. It's not like gases are continuously evolved. For bioreactor landfills, the stabilization period is about 10-15 years. For regular landfills, it could be 30-40 years. But Yeah, and the question is, okay, if it exhausts its capacity, what do we do? I mean, replacing the slag, it's impractical, So yeah, you say that we did our best, but main concern is the methane. Methane is being converted into low potent greenhouse gas that is still continuing. So get the benefit of whatever you can with the slag that you use, and then again, you are in a way getting rid of slag and reaping the benefits of it instead of sending it to a landfill. So yeah, the design life needs to be calculated, and we don't recommend it to be removed and replaced, but whatever benefit you can get, you get it, and then move on.
Then any other question?
As I mentioned, there are a lot of papers, we published, so feel free to download those, and then, any additional questions you have, feel free to contact me through email, and I will be able to, answer those questions.
Yes, thank you. Then
Thanks.
it's a good solution to contact directly to you.
Yeah.
Then thank you once again, your very interesting lecture.